neurocontrol. So brain, there's I guess two ways to kind of like see it. So first, if we are trying to we have to be able to read the code or the language of the brain, which we read with a bunch of like electrodes and stuff, and we can kind of intercept those signals and see like what's like happening. We can try to answer this like questions of how does this brain allows us to like see and like make decision, like think and take like and have actions. And so then we can kind of like control the brain, or not as in like mind control, but like control. So we can like communicate with the brain or tell the brain to do something. So in the case we can like if there's an epilepsy patient, like when they're having like seizures, like can we like give some sort of signal to kind of like stop the seizures in some sorts. It's really complicated because if you can think of it as like conversation where we're listening to each other, and then we see the facial expression and the tone, not just the word, but like the tone and the context and the body language. It all matters, and where we have this like continuous interaction in real time, and it's kind of like really complex thing that we kind of take it for granted. And if you're trying to do similar things with the brain, it's just and it's really difficult. So in order to compete that or fight that, they made this like brain initiative like 10 years ago in like 2012 or something, to put like a bunch of money in trying to like get a more understanding of the brain. And this is kind of like a moonshot of our generation in understanding the brain, but understanding the brain is really difficult. It's more of the more we like research and study, the more questions comes out, and the less we know, or we discover that we don't know as much as we thought we did. So we're kind of different goals would be to reverse engineering, or more achievable but still aspiring goal would be to reverse engineering the brain. Can we make something that's similar to brain in terms of hardware or software that and then we can say that we kind of understood the brain in this sense. So that's what human brain project is all about. And now controlling the brain, but it isn't really about like mind control. So we can control sensory perception, movement, memory, attention, emotions. So for sensory perception, just like the Matrix, can we like plug something in our like head, and then we can like sense it, like because we don't have to. We're not seeing it looking at the screen. We are like actually sensing it, because that's how we like sense the signal from like our eyes and ears, and like the smells and the temperature, the wind, everything, and we will be able to feel just like we are at whatever the place in the event. Movement, memory. It would be great if we can like improve our memory just by taking a pill, or have this electrical stimulation in certain parts of the brain, and it just like makes our memory like 10 times better, stuff like that. And it comes on and on, but it kind of becomes a little like we're going into more ethically gray area here. But to be careful about it, and we can also always use it for treating diseases and injuries. So go a little more into the circuitry of the brain. There's a lot of different circuits: decision making, motor learning, sensory perception. Also like the different diseases or malfunctioning of the brain, like Parkinson's, Alzheimer's, depression, epilepsy, traumatic brain injury. And there's no really a good cure for it at the moment. It seems like we're just treating the symptoms, not the actual cause. And that's why our treatments for Parkinson's and Alzheimer's and all this are not that great. And DBS is kind of working, deep brain stimulation, but then again, we still don't know why DBS works. And the last past decade, with the improvements in the measuring tool for our brain's activity has been exponential and explosive. So we temporal dynamics, so with temporal resolution of so electro, we can get signal as low as like 1 milliseconds resolution, and then for like hours. But it's hard we can't capture the whole person's lifespan with this like electrode. And similarly, like with scale-wise, we have a really small resolution or small scale, so we can see all this detail, but to get the bigger picture, we can't really capture the whole brain's activity. Whereas at like MRI, we can see the whole thing happening, but the resolution is not that great. Whereas also it's easier to see, like observe person's like lifespan with like MRI studies, like tracking, keep studying. And there's a different, like all the different method that kind of covers this map. So a lot of times we use multiple methods to kind of cover different resolution in terms of time and space. So there's just a bunch of examples. And it seems if you're just looking at this number of simultaneously recorded neurons, and this is not just like we say the MRI and then we can see the technically whole neurons, but the resolutions that are good enough that is to be efficient or effective. So we're looking at like an electrode or stuff like that allows us to record simultaneous individual neurons. And we see this almost like a Moore's law in semiconductors, which is like the number of transistors on a chip. We now see like almost like now we're going to like hundreds of simultaneously recording neurons, and it's doubling like every 7.5 years, it seems. Just different example. And so for treatments of different like Alzheimer and depressions and epilepsy, like they take like oral drug and pill, but it doesn't work in such a fast scale. It's not like we give some stimulation through a drug and then it comes out of the body in another like minute. It's not like that. But optogenetics provides a very effective a method for fast manipulation. So there's this protein from different like creatures that are really sensitive to like light, like from the deep sea or something. And then we do some genetic engineering, and we can put in like a virus, and then like put in the cells and like kind of like manipulate it. And if we have different like channels, where this one is so this protein is really sensitive to this like blue light, and then you shed the blue light, it like make them come this like a positive ions into the like cells, which they makes the like fire and it's so excitatory. But on the other hand, we can do the opposite, where negative ions are like coming in. So now when they are exposed to yellow light, so you hyperpolarize it, and then like so this would be inhibitory, and like other methods. The good thing about the optogenetic is that with electrical stimulation, we're exciting everything, right? But we can actually only excite it. But with optogenetic, we are able to like inhibit it as well. Also, as I said, electrical stimulus stimulates everything. So there's no like specificity in terms of a cell that gets excited. It's just a regional, right? But with optogenetic, we can be specifically excite or inhibit a certain type of cells, which is achieved by the delivery through the viruses like AAV, and we have more control over it in that way as well. And because they're only stimulated by a certain type of light, blue, yellow, or some others, what we can do is we can mix them up so that we can have like blue and yellow light, like a light in the skull, and then we can just like shine it and like independently and even at the same time, and we'll be able to have like greater control without interference. So it has a main advantage over just electrical stimulation, and then it's just a different time scales of like how fast they get excited and how the peak activations level. So this arch, this allows us to like just silence the whole cortical neuron that are mediated by arch, and then there's this halo rhodopsins. So like by optogenetics, there's the current injection, and they get like stimulated like that. So when there's a slight we get like just like inhibition. So when we put both of them, actually like it's able to really like inhibit quite well, like in both like scales, and it's like small time scale, and it just really works well. And this is a channel rhodopsins in vitro. And then with the blue light pulses, like we have so much control in terms of like time, like but they're really cool, but they're mostly just done in like in vitro, so like on a petri dish, on a brain slice or neuron cells. So it doesn't directly translate to in vivo, like in our body, like for human. Like apparently there's a lot of technical challenges, even just to like a non-human primate as well, for optogenetics to be useful. But this is on the rat's brain, so we have cannula, it's just like a port on the brain that we can use when we want after installing them. So we can like put optical fiber and then get light, and we can give light that way. Sometimes we use a strong LED nowadays because it's like works well enough, and then or you can just directly put LED here. So we can inject this virus that has this like optogenetic or photosensitivity and ability. So and we inject it in this like area called VTA, and they like projects it to like different area of the brain. And so by stimulating on the cell bodies in this VTA region, like we also kind of like influences like this area as well, and also like these other areas that like it projects it to. But if you do it on this like only this region, and we are selectively stimulating these neurons that are only that are reaching only to this region, rather leaving out this other neurons that's projecting to different area. Different things we can do. So this is similar ideas, but that the catch is that they both need to be present for it to be light stimulated, so that we can. In this way, we are also able to stimulate only the cell that's located here, that's projecting to this area, or this is the virus that and then enables the photo stimulations and so we put it here, and then these cells are like, now they're transmitted with this special gene, and then like we shine it over here, and then now we're also able to selectively just stimulate the neurons over here that are projecting to here. But it's just a multiple different ways of doing it, but we definitely get more resolutions and control over what we stimulate. So there are two parts, like the actuators and the sensors, so that we can think of it as an input and an output, or we are writing the neural code here, and then we are reading the neural code here. This is just a picture that's available over the counter. Like now, instead of just like electrode reading, can we like also install LED, and then like it also we can use it as a like stimulator and like alpha reader. And there's optogenetic stuff here. A lot of different like put electrode and get a bunch of signals from here. But it seems like this issue is still over like on this actuator side, that we have a little less control over here than the sensing side. So the control, so we have all these data and like tools. So like how do we put this together in a meaningful way? So we can learn from our like control of our nervous system itself. So oculomotor control is like our eye movement, right? And our eyes are like really cool. We have different muscles attached to the eyes that like allows us to move it. And also like the functions of this is that where this can be useful would be where there's like a predator coming like to us, and we have to like focus our like vision to that area, because we're not like if you're trying to read a book from just your peripheral vision, you can like see it, but you can't read it, right? So you have to like quickly turn your eyes. Another example is like when we're like moving our head and stuff, like we can actually still read stuff because our eyes are compensating our head movement with this, like oculomotors to be able to fixate our focus on one thing. So that'd be useful in general when we're running or walking or just any motions. Another example is just like this. They have this circuitry, so like your like focus is like staying still while your head is moving. You can like this have this like, control movement and the theory. So they like made a like whole like control system out of this old times, like in 60s or something. So there's a head velocity and there's like eye velocity here, and then you have this like motor neurons and eye neurons over here, and then eye neurons like speed out the negative G, the gain. So it's like allows us the eyes to move in the opposite direction from the like the motor neurons from our head, basically. And then, like if you look at how we like look at this like pictures and we track the eye movements, we get this like thing like we're focusing on the like primary features like eyes and like mouth. It's a really cool thing. If you're even when we're reading, like Jerry's usually like quite grouchy until he has had his morning coffee and read the papers. And when we're like reading it, like we kind of go like back and forth, like to ensure that like okay, after we reach the last word, we go back and ensure like yeah, we're done, or something like that. And then like for just random pictures, so if you're just like looking at pictures like without any instruction, we just look at things, you know. But when we like over here, but like we were told to okay, from this picture like find the trash bag, and then we have this like different specific pattern that they're looking for the trash bag. And whereas the free viewing eye movement, we kind of like go into different places, like we don't like think of it consciously, but like in the back of our mind, like under the hood, that we don't really go into same places as often to kind of like I guess maximize the information we get from the pictures or something. So they did a similar like control system, retinal error or distance between foreview and target, burst general motor neurons, etc. blah blah blah. And you can kind of like put it here. They made this like complex system out of it using this funny, rather simple equation, but complex. So like smooth pursuit. When we were like playing sports and then like balls are coming at us, like we are able to like predict its movement and like kind of following along. And it involves like a lot of different parts of the brains, and it's really incredible on how fast we can process information while it involves so many different parts of the brain to kind of communicate informations. Similar thing. Different example is like the roaches are really good at like maintaining certain good distance while moving very quick. So antenna here is like a J shape and making a contact with the wall and maintaining optimal distance, which is kind of similar to this like curve filler that was like popular apparently like 1960s, 70s when so like they probably get their wall positions with their like antenna, and then they like sense it with like mechanically, and then like goes to their brain, and then like rental certain order to the body to like do things, and that they can position them in a certain distance. The knifefish, they really like to like hide in like some spaces to like, you know, to evade getting eaten by predators, and then so we can like we can move this like apparatus around, and then see how fishes are able to like maintain their positions within the apparatus. And they use vision and electrosensory, and then like moth, fly stabilization. So they rely on visions and mechanosensing, and so like, and they use adjust their position with their like abdomen, wings, and like heads, angles, and like, etc. So it's another control system, like arm movement too. Our like arm itself has like sensory movement. Like we know where arms are like at all times. So we probably like select a certain task that we're gonna do, and then there's an optimal way to do so. And we do like have our like lifting a cup or something like that, and we get like sensory feedback from it, from like our tip of our finger, how the pressure, the weight, how much of force we have to exert, torque, etc. And then we get some noise, but it's meter and goes back to like this feedback loop.